All right, dear students, today we're going to be talking about the DNA. And also we will approach the RNA, which are the ribonucleic acids. So let us see this topic. All right, so as you can see here, we have the nucleic acids. So nucleic acids are going to be two, okay? So, first up, we got to see DNA as the genetic material for living organisms. Okay, and DNA is going to be called DNA because that is an abbreviation that means deoxyribonucleic acid. Okay, and it's this genetic material that is found in all living organisms. So all living organisms will have it from a single plant to the most complex living organism that can be, for example, a human with different systems and so on. Okay, so the RNA means ribonucleic acid, and it's also present especially in the cytoplasm and ribosomes that are a part of that are parts of the cell, right? So remember that the ribosomes can be found in the cell as an organelle, and also we can find the DNA in the mitochondria, right? Okay. So then we have to see that this is the universal genetic code. So the DNA is the same for all species, making it a universal language for life. So thanks to the DNA, we can detect, for example, presence of living organisms where or when they have been. Okay. Viruses in RNA. Some viruses like SARS-CoV-2, which is the famous COVID-19, use the RNA as a molecule or substance that will transport their information, and at the same time they will need these nucleic acids to affect the organisms that will contain them, right? So we have, for example, viruses like influenza, the dengue, or polio. Okay, and their location in cells is basically found in the chromosomes. There are these famous X structures that are going to be found um, inside the nucleus, but also you can find the DNA in chloroplasts and mitochondria. Okay, the RNA is mainly found in the cytoplasm, but I want to tell you that the DNA depends in certain way from the RNA, okay, specifically speaking from the messenger RNA that will transport some information from the nucleus to the cytoplasm in order to code that information in proteins. And that is one of the main findings in molecular biology, and that is called the molecular dogma, right? That is known as central molecular dogma of biology. Okay. Okay, so we have that the DNA contains some monomers, nucleotide structures, right? And the DNA consists of four bases, such as the adenine, the thymine, the guanine, and the cytosine. Okay, and the RNA is going to be the same, but as you can find here, for example, in this image, we're going to find a thymine. All right. Instead of that, you will find uracil. Okay. Okay. So maybe up to this point, you're wondering why does the DNA have a different name to that one that appears in the RNA? So what is the main difference? So I would tell you that one of the main differences you can find here is that in the third carbon, the DNA does not contain an oxygen. Okay, that's why we call it the oxyribonucleic. I mean, the oxy, no oxygen, right? So in the case of the RNA, what you can find, for example, is that there will be an oxygen atom included in the pentose sugar, but that's something that I will explain you after. Okay, so here you can find, for example, the 3 different types of RNA. Okay, and this is the cell. And inside the nucleus, as I had told you, you will find the DNA. Okay, and in certain way, it depends from the RNA. Okay, so we have 3 different types of RNA. I will write them here. I will type their names here. Okay. So the types of the RNA that you will observe are the messenger RNA. That is represented with an M, right? RNA. So this is an RNA that will travel from the nucleus to the cytoplasm, as you can observe here. So we will have different types of RNA. They are called the messenger RNA that will take the information from the DNA strand to the outer part of the nucleus, so it can travel throughout some porous here. So the nucleus contains some porosity, and it will travel to the cytoplasm in order to take that information out. Okay, but also we're going to have something that is called the transfer RNA. Transfer RNA, and we're going to have something that's also called the ribosome RNA, right? Ribosomal. Okay, so as you can see, the messenger RNA will participate in the transcription process. We also have the messenger RNA processing processing the message, right? So then the message has been processed, and it will take out to the cytoplasm, right, throughout the nuclear pore. Okay, there is a concept about the unity of all living organisms. They will contain nucleic acids, but also viruses are included in this equation. This universality of genetic code indicates the genetic connectivity of life on Earth and explains how viruses can all take over and use the biological machinery of cells in order to act out as if they were parasites. Obviously, they are not parasites, but they behave in a certain way. Okay. So as you can see in these 3, we have the root of 3, and here is supposed to be found something that is really famous in biology. It is LUCA. LUCA was the first organism that gave origin to the other ones. Okay, so we have the root of 3. We have the orangutans, chimpanzees, the Neanderthal, until the modern humans, Homo sapiens sapiens, right? So also we have the cheetahs here, gorillas, and so on. Okay, all living organisms and viruses contain nucleic acids. This universality of genetic code indicates this interconnectedness, right? Okay, so it's crucial to talk about the DNA and the proteins in the nucleus. So we had a process here, and here's where we come up with 2 great scientists that were Friedrich Mishael and Levine, right? So Levine was the person that understood how these substances played a role inside the nucleus, right? So the DNA, in the early 1900s, proteins were considered better candidates for transmitting hereditary information. So in that way, we didn't know about the DNA or the RNA, but we also considered some proteins as those candidates that could transmit that genetic information. And then we had Friedrich, Mishael, and Levine, and they made significant contributions to understanding DNA and proteins inside. Okay, so in 1869, um, Friedrich discovered a substance that was called the nuclein, right? Now known as the DNA. So now you can see that the DNA is that famous substance, right, inside the nucleus. But he suggested that it was important for heredity, but unclear how it worked. Okay, so in 1909, Levine identified nucleotides as the building blocks of DNA. So when I say nucleotides, I mean that these scientists in certain way identified what was the sugar phosphate, the sugar phosphate group, what was the nitrogenous base that was contained in these nucleotides, and also was the pentose sugar, right? So also was the nitrogenous base, and finally the phosphate group. Okay, so but he made a mistake, and the mistake was incorrectly taught the DNA had simple repeating structure. So currently we understand that we can have the DNA, but it does not repeat its structures. Why? Because the nitrogenous bases can variate. So you can have, for example, a single strand of the DNA because it contains two, but I'm going to take one to exemplify this concept. So let's suppose that we have adenine, cytosine, a guanine, then we have guanine, adenine, thymine, and he thought that all the DNA structure was the same, but it was not like that. So what happened was after certain experiments, after the famous girl of the DNA, whose name is Rosalind Franklin, and that was the scientist who took the photo to the DNA, and Crick and Watson, or Watson Crick. Basically, they drew the structure of the DNA, illustrating it in detail now, and they discovered also that the nitrogenous bases weren't the same, right? But they variated, right? So that was his mistake. Okay, proteins were still considered to carry hereditary information until later. Okay, and we had something that's called falsification of early theories. So Misher and Levine's ideas were important but not complete at all. Later research were taken by Avery, Hershey, Chase experiments. They confirmed that the DNA was not a protein, but it was a genetic material, right? And Levine's model of DNA structure was refuted by the scientists that I had previously mentioned in 1953, when they represented the DNA as a double helix structure. Okay. Okay, so here there is a video. If you want to go and watch it, but I will continue. Okay. Okay, so the components of the DNA. The nucleotide consists of a nitrogenous base. Okay. And that nitrogenous base can be adenine, can be thymine, that you can see, those letters you can see here are only the initials. I didn't write all of them in a full way because I considered it was not like, okay, in certain books we manage this as if they were just initials, and that's okay, the reader understands. Okay, so we have the adenine, thymine, cytosine, and guanine. And then, okay, the nitrogenous base is normally represented by this squared that you can find in purple color. Okay, so the green color represents the sugar pentose. Okay, the sugar pentose, as I explained, my students means pent means 5 carbons, right? So we have each one of these carbons is represented by one of the corners that contain this pentagon, right? Pentose from pentagon, so it means 5 sides. So in the third carbon, it will not contain the oxygen carbon, right? In the third carbon, the oxygen ion, sorry. Okay, so they can be, for example, this base can be adenine, thymine, cytosine, guanine, and the pentose sugar is the oxyribose. And finally, in the circle, there is Somewhat like pink is going to be the phosphate group, okay? Really important in the directionality of the DNA. Okay. So it contains 4 liver nucleotides to form DNA strands. Really important to understand that the nucleotides basically assembly themselves by using condensation reactions, and that is what at the same time allows that the DNA coils, right? And it is one of its super skills. Okay, so this is the chemical representation of the sugar, pentose, the base, nitrogenous base, and the phosphate group. Okay, DNA extraction planning method. So there are some steps for DNA extraction from plant or animal tissue. Remember, you can extract the DNA from any single living organisms you can find. Obviously, you got to break the cells. So you heat it and smash. I mean, when I say heat, I mean the sample of the living organisms you've taken previously. And dissolve membranes, so you can use detergent to dissolve lipids, for example. And why do we dissolve these membranes? Because we need to access to the DNA that is contained in the nucleus. Separate DNA. So then we're going to add salty water and ethanol. You can also use isopropyl, but okay, so in this case, I recommend you to use ethanol to precipitate DNA. So when we precipitate the DNA, it will present in the test tube, okay? It will present somewhat like 2 phases. So in the less dense phase, you will find the DNA contained in on the surface or in the middle of the surface, right? So DNA is soluble in water, but not in ethanol. And this allows the separation. Okay, so there is a link that I would like to show you. Okay, so let's go to this link, right? I hope you're comprehending what I explained. Okay. DNA is extracted from human cells. Okay, so we're gonna play this simulation. Start the lab. DNA extraction by the techniques virtual laboratory. Okay, so here, as you can see, we have the DNA, right? Why, you may ask, do you need human DNA? We can test some DNA in a crime scene, let's suppose. Okay, so here we're going to apply the genetic testing, right? So this can be also used for body identification or analysis of forensic evidence. Scientists isolate DNA for a variety of reasons, some of which include genetic testing, as you saw, body identification, and analysis of forensic evidence. So let us approach to this subject. DNA extraction is typically the first step in a longer laboratory process. DNA is an important part of that process because the DNA first needs to be purified away from proteins and other cellular contaminants. So we need cells because that's where the DNA is inside almost every cell in our bodies. There is a nucleus, and inside each nucleus, right? So I mean, all nuclei are about 2 meters of DNA. Let's see what's next. So this is a cell, and inside we have the nucleus, right? And we're gonna have the DNA packed in chromosomes. At the same time, it will be attached by some proteins that are called histones. And this is the DNA. And there are the nitrogenous bases, okay? So where do we begin? First, we need to collect some cells from our test subject. Okay, let's see. So I'm going to extract some cheeks cells. Okay. Okay, you can use um, your mouth that constantly loses skin cells every single day. Uh, and these are cells we're after. Okay, so then once you have taken those cheek cells, we have two bar cells open to release DNA. So the first step was collecting cheek cells, no? So once we have collected the cheek cells, you got a bar cells open to release the DNA. And then you separate the DNA from proteins and the rice. And finally, you isolate the concentrated DNA. So let us see. So here are the materials and equipment you will need to purify DNA from chick cells. So we have the warm water bath, the centrifuge to separate the mixtures by densities. We have the buccal swab, sample tubes. We have some substances. Okay, let's get started. So the buccal swab, and you insert and drag the swab over the inside of the test subject's mouth to collect the sample. Say, let's see. So inside this, you will find different cells. They are called cheek cells. So every single part of your body contains cells. Really crucial to comprehend that, all right, in many cases in the laboratories, we used the cheek cells because they are easily taken by this buccal swab. But you can also obtain cells from different parts of your body, right? Included your blood. Okay. Okay, so that is not a part of substance, but okay, I mean. So next you will place the swab into an Appender tube. Look closely at the swab and know that it is covered with hundreds of tiny cheek cells, the ones that I show you in the last slide. Inside each cheek cell is a nucleus. And inside that nucleus that we saw in the introductory part, you will find the chromosomes, and the chromosomes are these packs that contain the DNA. Okay, so then we're going to take the sample inside the tube. Okay, we'll extract the lysis solution, right? So the end of the swab must be cut off so you'll be able to close the tube using the micropipette up some lysis solution to the tube. Lysis is a Greek word that means to separate, okay? Okay, so we'll take the sample, then I will put it in a warm water bath. Place the tube into the warm water bath. Okay. So then our substance will be having some physical changes, right? And those physical changes are that will break the membrane, right, to access to these histons. Okay, so the lysis solution you just added contains two important ingredients, detergent and an enzyme called proteinase K. Okay, the detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones, and the proteinase K cuts apart the histones to free the DNA. Okay. So the cells have stayed in the warm water bath long enough for the DNA to be freed from the cells. We have removed the swab from the tube. Add some concentrated salt solution to your tube. Okay. So I'm going to take the pipette. Already taking the salt. Okay, so molecularly speaking, you will see the DNA, the debris, and also the proteins, right? So the salt causes proteins and other cellular debris to clump together. Okay, and then we're going to separate these using the centrifuge. The centrifuge separates mixtures according to their density. So the denser, the more dense, because we can also say that in size, the denser and more dense is the scientific vocabulary, are going to be placed or laid down, right? And those ones that are less dense will be placed on the surface zone. In order to balance the centrifuge, a tube containing water is placed opposite your tube. Okay, so we got to place another tube. We're not going to use that second one, the second tube, but it's in order to balance the centrifuge. Okay, so you close it and let us wait for some minutes. Let's suppose that some minutes have passed, and then we'll have the proteins and debris on the lower part of this sample, right inside the centrifuge. The tubes spin around at high speed. Heavy clumps of proteins and cellular debris sink to the bottom of the tube, while the strands of DNA remain distributed throughout the liquid. Okay, so let's see what's next. Right then, use the micro carefully to remove the top liquid, which contains DNA, and place it into the clean tube. So the proteins and other cellular debris stay behind, stay up behind. Okay, so that's it. And then we are in the fourth step, which is the isolate concentrated DNA, and add some isopropyl alcohol to the tube, right? In this case, we're using isopropyl, not ethanol. Okay. And then what we'll have is the DNA, right? Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble, does not stay dissolved in isopropyl alcohol, it comes out of solution. You can now see the clumped DNA with your naked eye. And that's how it works. Then I place it again into the centrifuge. Okay. Close it. Right, close it, open it again. And I will have the DNA extraction process complete. So once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years, or you can move on to your next experiments you have just extracted. Try your hand of DNA extraction using common household items you can find in your kitchen. So this is an experiment that we have made with students several times. It's an amazing experiment that you can practice by yourself, obviously under adult supervision or under a professional supervision. Okay, so let's see what's next. Remember the components of the DNA, right? There are nitrogenous base, pentose sugar, phosphate group. And the nucleotides are linked via condensation reactions for different nucleotides for DNA strands. This is what we do, what we did some minutes ago. Simple diagram of DNA component using simple shapes to represent it. phosphate group is represented with a circle, pentyl sugar pentagon, nitrogenous base with a rectangle, and you can illustrate how they are connected, right? We're going to review some key points. Early researchers like Mishael and Levine contributed to the DNA understanding, but modern discoveries corrected their theories, placing the DNA as a stat that could contain the genetic information instead of proteins. DNA structure is now known as double helix structure, and that is made of nucleotides. And finally, DNA extraction methods can isolate genetic material for study. Okay, so in diagrams of nucleotides, you use the circles, as I told you, the pentagons and the spheres, okay, to represent or and the rectangles are represented, right? So we have the phosphate group, the pentyl sugar. Notice here that in the pentyl sugar here, in the third carbon, we have a hydroxyl group. And sorry, in the fourth carbon, we have the hydroxyl group, and in the third carbon, you will not have oxygen. But in the RNA, that is the ribose, will have in the third carbon an oxygen, right? That indicates that it contains oxygen. The oxyribose does not contain the oxygen. So we have the adenine, guanine, thymine, cytosine, four nitrogenous bases, important for the DNA. The condensation. So in this case, we are going to condense the DNA, and when they condense, they will release water molecules, right? That will bond together. Okay. The bases can be divided into two groups: the purines that are adenine and guanine, and pyrimidines that are cytosine and thymine, based on their molecular levels. We have the cytosine, guanine. This is another representation of what the DNA is. Okay. Okay, so the sugar phosphate group, the phosphate backbone of DNA and RNA. So is this structure you can see in the middle in green color, right? And it's composed of a sugar phosphate backbone, which is uniform and strong, forming a continuous chain. The nucleic acids, that is the orange. I studied from the middle because we were talking about sugar phosphate group, right? But the first one that is nucleic acids. They are large linear macromolecules made of nucleotides. Examples are DNA and RNA that is shorter. And the blue one that is the function, right? And it holds genetic information that dictates protein synthesis. This is related to the central molecular dogma, right? So the sugar phosphate bonding in nucleic acids. So we have the backbone alternating sugar and phosphate molecules, the bonding, and the types of bonds that will use the DNA are covalent bonds. Remember that covalent bonds are given between non-metals and non-metals. In this case, we're using a lot of hydrogen atoms, right? And they are stable backbones in both DNA and RNA strands. And finally, the condensation reactions, nucleotide bond throughout condensation, building long chains of nucleotides that are going to be called polynucleotides. Okay, so nucleotides become chemically combined together, phosphate to pentyl sugar, bicovalent bonds with a sequence of bases attached to the sugar residues. So up to 5 million nucleotides condensed together in this way, forming a polynucleotide that is called finally as nucleic acid. Okay, so the bases are these ones, right? So you can have, for example, adenine, guanine, thymine, cytosine, etc. And nucleotides are added at this of the growing polynucleotide. The RNA polymers are similar, but in this case, instead of having the Thiamine, we are going to use uracil. That is this last nitrogenous base you can locate here in this drawing, right? So important to understand that the phosphate group is this one, and it conforms an nucleotide, right? So we have the adenine, the nitrogenous base, and the pentyl sugar, right? Then we have cytosine, guanine, and uracil. But as you can observe, the RNA does not contain double helix. It contains one single strand, or you can also say is single-stranded. Single-stranded. That is the word we use in biology, right? So structure of RNA, single-strand structure is usually single-stranded, unlike the double-stranded structure of DNA. Bases, we have the uracil, cytosine, guanine, adenine, and uracil. And the uracil will replace thiamine that is found in DNA. And the orientation, carbon atoms in ribose are numbered from 1' to 5'. Okay. So in this case, we're going to name these carbons from 1, 2, 3, 4, 5. And in 5 carbon, the 5', you will obtain the phosphate group. But in the other strand, it goes from 5' to 3'. So in one single strand, we'll have the directionality of the DNA from 3' to 5', but in the other one, we'll have from 5' to 3'. And what it means is that these two structures are complementary, but at the same time, they are not in the same direction, right? So this is due to some processes that I'm not going to explain you a lot today because this is not the class about. Maybe in a molecular biology class we can talk about it. But what it means is that they are complementary, but they are at the same time inversely placed. Okay. Okay. So processes that I mentioned, right, are, for example, the replication, the transcription, and the translation, right? And those processes are taken mostly by the RNA. So in certain way, we can say that first we need the RNA to code the DNA and to translate that process into proteins that will help, into amino acids that will help us to have proteins that are so important to make our organisms work. Okay, so the DNA replication, the purpose is to create an identical copy of DNA before cell division. So there are some key steps. The DNA unwinds and separates. So you can observe that the helicase, that is an enzyme, will be breaking the hydrogen bonds of the DNA, right? And at the same time, we have another enzyme that is the DNA polymerase. And what the DNA polymerase will do is basically that it will make stable this single helix that at the same time is isolated, right? And okay, so this is the original strand, DNA strand. And we have the DNA polymerase acting in both directions it just acts in one single direction, sorry. Okay, but it's acting in both helixes. Okay, so the result is two identical DNA molecules, as its name says, replication. So what it does is basically replicating the nitrogenous bases by using the sample of nitrogenous bases. Okay, as you can observe here. So in this case, for example, guanine is all the time bonding to cytosine, okay, and thiamine is all the time bonding to adenine. So the DNA polymerase detects that, and what it does is basically that it will place the other lacking nitrogenous base in this case. Okay, in the transcription process, that is this one. The copies of DNA are going to be taken into by the messenger RNA, mRNA, right? And the key steps are RNA polymerase binds to DNA and synthesizes the RNA. The messenger RNA is created as a complementary strand, so it will be traveling from the nucleus to the cytoplasm in order to take that information out. And the result is that it will carry genetic code from DNA. And finally, we have the translation. And in the translation process, the messenger RNA is read by ribosomes. And here we'll have another type of DNA that is called the transfer RNA. Right, in the transfer RNA that is represented with the letters tRNA brings amino acids which are linked into polypeptide, the structure you can observe here, and the result is a protein synthesis. So each one of these single circles represents a nitrogenous base. So if you take 3 of them, for example, what you will obtain is a codon. So a codon can be 3 nitrogenous bases grouped, and that will translate into an amino acid. Okay, so when you take the amino acid and you take three of these nitrogenous bases, their combination will produce an amino acid. And we have a total of 64 amino acids that will code into proteins in our body. I will explain you that. So the directionality of RNA, we can identify direction or polarity in the DNA double helix. The phosphate groups along each strand are bridges between carbons 3 of one sugar molecule and carbons 5 of the next. And one chain runs from 5' to 3', while the other runs from 3' to 5'. So they are complementary, but at the same time, they are inversely placed. Okay, the existence of direction in DNA strands becomes important in DNA replication when DNA is copied, the replication of DNA, and when the genetic code is transcribed into messenger RNA, a process called transcription. And when the message encoded in the messenger RNA is read to 4 proteins, a process called translation. Information in the DNA lies in the sequence of the bases, cytosine, guanine, adenine, and thymine. And the sequence takes the order in which specific amino acids are assembled and combined together. The code lies in the sequence in one of the strands. The coding strand, the other strand, is complementary to it. It is a coding strand that becomes a template for transcription. The coding strand is always read in the same direction, in the 3' to 5' direction. Okay. Okay. Question: is read by the polymerase? Okay. Let us continue. A single-stranded molecule of RNA is formed by complementary base pairing. The RNA strand is synthesized in the 5' to 3' direction, and the messenger RNA are translated to the 5' to 3' direction into amino acids by a ribosome to produce polypeptide chain. The details of these processes will be explored in the subsequent chapters, but in this case, I'm not going to approach this. So there are 3 types of RNA: messenger RNA, tRNA, rRNA. Okay, the messenger RNA is the one that carries the information. Is one from the nucleus to the ribosomes. The transfer RNA is this one that brings amino acids to ribosomes during protein synthesis. And the ribosomal RNA, that is this one, is the final that is structural component of ribosomes where the proteins are assembled. The formation of RNA, it forms throughout condensation reactions. Okay, the nucleotides of monomers condensate. The 3' to 5' phosphodiester bond links the phosphate group of one nucleotide to the 3 carbon of the next nucleotide sugar, creating backbone. Okay, and this is the RNA codon table, as I told you previously. The amino acids that are these ones that appear here in an abbreviated version. For example, if you combine guanine with uracil and with another uracil, you will obtain valine. Okay, and this is like a multiplication table. For example, if you combine adenine with uracil, I don't know, with guanine, for example, and also with uracil, you will obtain methionine. Okay, that is MET, methionine. Or, for example, if you combine cytosine with uracil and with adenine, you can obtain lysine, right? That is represented with LEU, and so on. And each one of these amino acids will code to 4 proteins, and the amino acids are the building blocks for life, for proteins, right? So we have different types of amino acids. I'm not going to go deeper into this topic. They are, for example, alanine, arginine, asparagine, aspartic acid, cysteine, and so on. Glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine. Threonine is really important, and then we'll see why, but not in this video. Tryptophan, tyrosines, valine, and so on. Okay. Okay, so important to comprehend that we have 1, 2, 3, 4, 5. 5 times 4, 20 amino acids. Okay. And some of them are essential. And okay, but we have a total of 64 codons, right? Because if you multiply 1, 2, 3, 4, 4 times, it contains 4, right? 4 times 4 equals 16, and 16 times 4 equals 64, right? 16 times 4, 24. 4 times 1, 1, plus 2, 64 different combinations. Okay, polymer, a large molecule made of repeating subunits. Those subunits are monomers. A codon, that is a sequence of 3 RNA bases that specifies an amino acid. Uracil, that is a nitrogenous base in RNA that pairs with adenine. Okay, those are the key terms. So this is what I previously explained you. Okay, the RNA is formed by the condensation, and remember that the DNA is a double helix structure, okay? And it has an anti-parallel orientation, as I told you. So it's complementary and at the same time anti-parallel, right? So one strand runs from 5 prime to 3 prime, while the other runs from 3 prime to 5 prime. And between these nitrogen bases will have the hydrogen bonds between the complementary bases, and they will be 2 hydrogen bonds or 3 hydrogen bonds depending, right? So here we have the famous Watson and Crick, but I don't want to talk about them before talking about Rosalind Franklin, the one that took the first capture of what the DNA was. Okay. So physical model making helped Watson and Crick to establish the structure of DNA in a number of ways. It allowed them to combine what was known about the chemical content of DNA with information from X-ray diffraction studies previously established or undertaken by Rosalind Franklin. By building scale models of the components of DNA, that were able to attempt to fit them together in a way that agreed with data from other sources, such as Chargaff's rules. They made several arrangements of the scale model until they found the best one that fitted all the data. So it was like structure. Right, I want to tell you that they first presented their model using some cardboard paper, and they had to modify it because they discovered it was mistaken. But okay, so basically it was it, yeah. Base pairing in the DNA, complementary base pairing. We have the adenine pairs with thymine, and it contains two hydrogen bonds. You can see the hydrogen bonds representing with blue color here. Okay, but in the case of guanine that pairs with cytosine, it will have three hydrogen bonds contained here. Okay, so the double helix twists, the structure twists 360 degrees every 10 nucleotides. That equals 3.4 nanometers. Okay, DNA modeling and discovery. So modeling science simplifies complex biological systems. Watson and Crick in 1953 created the first model of DNA structure based on X-ray data. And the role of models used for predictions, calculations, and refining new data emerge. Okay, applications and impact of DNA knowledge, genetic fingerprint, for example, fingerprinting that is used to identify individuals, paternity, and criminal evidence. Also, we can find the applications in medical advancements, such as the gene editing, CRISPR, that is a technique, okay? The molecular genetics and monoclonal antibodies. And but also we have some ethical considerations, right? So the genetic engineering basically has raised some concerns in the scientific community that basically criticize certain advantages that some individuals can have according to the DNA modification. Also, we can design babies with certain phenotypes and genotypes, physical characteristics, and genetic conditions. Okay, but it also brings to the table some worries, some privacy concerns at the same time, and discussions to have debates, right? So that's still on debate, but currently it costs a lot of amount of money, but it is still a worry. Okay, so ethical and social implication of DNA research. The utilitarian ethics emphasizes the greatest good in decisions of DNA applications. Determinism concerns genetic explanations for behavior may impact social support for interventions. And the public policy impact, genetic data use raises issues with insurance, privacy, and societal inequality, right? So this is a scientist that we cannot skip for nothing. Crick and Watson brought together the experimental results of many other scientists, and from this evidence, they deduced the like, structure of the DNA molecule. But Rosalind Franklin, who is known as the forgotten woman of DNA, was a person that captured the DNA, right? So this is for the readers. If you want, you can stop the video. But what I can tell you is that she played a critical role in developing the first model of DNA throughout her work. With X-ray diffractions, she produced the key image, particularly the photo 51, in her article at King's College London, which provided clear evidence of DNA's double helix structure. And this image in her research helped identify the molecular spacing in helical form of DNA, and that is essential for building the accurate model. Despite her pivotal contribution, Franklin did not receive the same recognition as Watson and Crick because of how her findings were shared. And so in certain way, Watson and Crick they have been criticized because they didn't mention her a lot, right? And even though they were awarded the Nobel Prize in 1962, after Franklin had passed away, as Nobel Prizes are not awarded posthumously. Okay, so here we have some key concepts. The exercise to approach these key concepts, alleles, for example, um, chromosomes, genes. So we can say, so this is an exercise you can do by yourself. It's like an assignment that I can give, right? Okay, the conservation of genetic code. As you know, we have different kingdoms, the bacteria kingdom, archaea, eukaryotic kingdom. And okay, we have a common ancestor that is called Luca. And despite billions of years of evolution, the basic codon amino acid relationships have remained consistent with only slight variations. This conservation supports the idea of a shared ancestry and highlights the role of conserved sequences that are parts of DNA that remain unchanged over time generations in maintaining essential cellular functions. Okay, also, some conserved sequences are coding directly involved in protein production, but others are non-coding and have unclear functions. Okay, they remain there, but with no clear functions. So the genetic diversity and mutations, the four bases code adenine, cytosine, thymine, guanine in DNA, adenine, cytosine, uracil, guanine in RNA enables the vast diversity of life, allowing thousands of unique proteins in complex organisms like humans. So DNA mutations introduce changes in the genetic material, which can lead to variations in traits and contribute to evolutionary diversity. Really crucial to figure out that mutations are not all the time bad or negative, right? So mutations can be positive, that are led through something that is called natural selection, adaptation. Mm, but also they can be neutral. I mean that they're not positive, neither negative, at least until they are discovered to be. But we have also neutral mutations. At the same time, we have negative mutations, right? But not all the mutations are negative, right? Really important to comprehend that not all of them are negative, right? And the directionality of DNA, remember the 5 prime to 3 prime is basically the running direction of this DNA. And also goes from 3 prime to 5 prime in the other helix, right? So this anti-parallel structure is essential for DNA replication and transcription processes. In DNA sequence of bases, adenine, thymine, cytosine, guanine provides information to code specific proteins by dictating the amino acid order in protein synthesis process. Role of purine and pyrimidine bonding. Watson and Crick's model of DNA highlighted purine pyrimidine pairing adenine with thymine and cytosine with guanine, which makes the DNA stable. And the consistent structure across different sequences is crucial for DNA's role in storing and transmitting genetic information. So DNA packaging in chromosomes. In eukaryotic cells, DNA is highly organized and packaged within chromosomes. This is made possible by nucleosomes, which consist of DNA wrapped around histone proteins. And this structure allows for efficient storage and protection of DNA within the cell's nucleus. The idea of conserved sequences, structural insights onto DNA, and the packaging mechanisms underscore how molecular biology principles support life's continuity and complexity across diverse organisms. Okay, so we have, for example, here the 3 hydrogen bonds, the double hydrogen bonds, and this is the double helix. Okay, so this is the direction that goes, in this case, from 3 prime to 5 prime, and in the other helix from 5 prime to 3 prime. So they are complementary, but at the same time anti-parallel. Okay, but I have a question for you. What do you think that attaches, or, yes, what do you think that basically binds the DNA? And there are some proteins that are called the H1 histone. In this case, the histones binds the DNA to the histone bed, right? So, and this is basically a demonstration on how the DNA molecule is packaged in eukaryotic cells. DNA double helix wound round histone proteins bed called a nucleosome that is 30 nanometers of fiber, right? So the core of nucleosome of 8 histone molecules forming the bed structure. The DNA double helix wound twice around histone core, and the H1 histone binds the DNA to the histone bed. So this is a micrograph of the metaphase in the cell cycle, right? This is a chromosome, and this is the electron micrograph demonstrating that at this stage the chromosome is at its maximum condensed state. So the packaging of DNA in the chromosome, right? Okay, so the DNA packaging during the cell division, the DNA is tightly packed to form a condensed structure known as a chromosome. And this is achieved by a series of coiling steps. The DNA wraps around histone proteins, forming nucleosomes. These nucleosomes further coil to form chromatin fibers. Finally, chromatin fibers supercoil and organize around non-histone scaffold proteins to create a highly condensed chromosome structure seen during cell division. Okay, so it gives us a supercoiling advantage. So the main advantage of supercoiling chromosomes during cell division is to efficiently condense the DNA. That reduces the risks of DNA damage and tangling. And this tight packaging also enables the cell to manage and segregate chromosomes accurately, ensuring that each daughter cell receives a complete and intact set of chromosomes. And the molecular visualization, you can go to different online tools such as Protein Data Bank that allow users to visualize DNA and protein interaction at the molecular level. These tools help illustrate how DNA wraps around histone proteins, but we're not going to see it today, right? So we have the Hershey and Chase experiment, and this experiment demonstrated that DNA, rather than being a protein, is a genetic material, and they use bacteriophages. By tracking labeled DNA and protein in infected bacteria, both concluded that only DNA entered the bacterial cells, confirming DNA's role in genetic inheritance. So we have the electron micrograph of a bacteriophage infecting a bacterium. Okay, this is a type of virus we have. There are different types of virus, but this is a bacteriophage, obviously that is taken from a bacteria. And the structure of the phage is basically that the head of protein it will contain the nucleic acid, and this is the tail of protein. And okay, this is the base plate of protein, right? Okay, so in their 1952 experiment, Alfred Hershey and Martha Chase used a bacteriophage that infects Escherichia coli E. coli bacteria to determine whether genetic information was carried out in the protein code or the DNA of the virus. They designed their experiment with two key radioactive labels to distinguish between these components. And the steps were, okay, they were labeling DNA with radioactive phosphorus, 32 phosphorus, that is present in DNA but not in protein. So they labeled the DNA core of the virus with radioactive phosphorus. This ensured that any radioactivity detected in the bacteria would be from DNA if DNA were the carrier of genetic information. They labeled protein with radioactive sulfur, 35 sulfur, and sulfur is found in proteins, specifically in certain amino acids like methionine and cysteine, but it's absent in DNA. By labeling the protein code with radioactive sulfur, 35, they could trace any radioactivity detected in the protein if protein were the genetic material. Okay, the experimental processes and findings. The bacteriophages infected several batches of E. coli. One batch with the 32 phosphorus labeled DNA and the other with the 35 sulfur labeled protein. After allowing the phages to infect the bacteria, Hershey and Chase used a blender to separate the viral proteins codes from the bacterial cell. And finally, they centrifuged the samples in this machine that I showed you previously, allowing the heavier bacteria to settle at the bottom. Okay, and the results were that in the batch with radioactive phosphorus 32, The radioactivity was found inside the bacteria cells, indicating that DNA had entered the bacteria. And in contrast, the radioactive sulfur, sulfur 35, was detected in the remaining solution, not inside the bacterial cells, but in the remaining. So this meant that the protein did not enter. In the conclusion, they wound up this experiment demonstrated that DNA, not protein, is the material responsible for transmitting genetic information, as only the DNA from bacteriophage enter the bacterial cell and directed the production of new viruses. Okay, and this is the process. See the protein code, the DNA of a bacteriophage that enters the host cell and takes over the cell's machinery, so causing new viruses to be produced. Right, I hope you have understood many things on the DNA and comprehend all the processes we're taking here in scientific evidence. Right, see you in the next class, guys. This is Lingually. This is Mishael Wright, owner of Lingually and IC.